Hello, RNA world. Can everyone hear me? I would think so. Welcome to the RNA targeting technology session sponsored by Locanabio. I'm the Vice President of R&D at Locanabio. At our company, we're using RNA targeting technologies to treat chronic human diseases. To target RNA or disease-causing RNA, we use RNA binding proteins encoded in DNA genomes for viral and non-viral delivery. Today, we have an amazing lineup of 3 expert speakers in the field of RNA targeting. And these are Professor Oliver Rackham, who joined us from Curtin Medical School in Perth, Western Australia. Dr. Franco Pagani, he's the group leader of human molecular genetics at the International Center for Genetic Engineering and Biotechnology, Trieste, Italy. And Mitch O'Connell, assistant professor from the Department of Biochemistry and Biophysics at the Center of RNA Biology from University of Rochester Medical Center in New York. We'll have about 3 15-minute presentations with 2 to 5 minutes of questions. Please don't forget to pop in your questions in the Q&A box. So we can have a scientifically enriched hour. Without further ado, I would welcome Dr. Oliver Rackham to come onto the virtual stage for our first talk.
Thank you so much for the kind introduction, Ron. It's a real pleasure to join you from sunny Western Australia. And hopefully you can see my slides coming up now. So as Ron said, I'm a professor at Curtin University, and I'm going to talk to you today about building artificial RNA binding proteins. I'm also part of a Center of Excellence in Synthetic Biology and a scientific consultant for Locanabio. So we really take a synthetic biology approach to manipulate RNA binding proteins and create new RNA targeting tools. And the way I sort of encapsulate synthetic biology in a nutshell, because a lot of people say, well, isn't it similar technologies to other biologists? And in essence it is, but we have a different ethos. And I think this is sort of summarized well in this analogy that in traditional biology, you're able to study anything that exists in nature. Similar to if you're skiing, you can do any run you like within a ski resort. But anything beyond that is not possible. In synthetic biology, instead, we take inspiration from the natural world, and we, sorry, my slides have just dropped there. Pop them back up. We take inspiration from nature and instead trying to extend that to do new and useful things. And particularly in our lab, we're really interested in RNA. Of course, one of the most famous offerings from synthetic biology that everyone's aware of is CRISPR-Cas and TALEN enzymes, which have enabled almost any manipulation of double-stranded DNA to be possible in today's laboratories. But when I started my lab, we were really interested in RNA, as many of you are in this conference. And we saw a big gap that CRISPR-Cas and TALEN technologies were targeting double-stranded DNA. But as you know, there's a bountiful array of manipulations that happen at the RNA level, right from the beginning of transcription through to destruction of messenger RNA and the end of its life. And so we wanted to be able to hack into these by using different synthetic biology tools. And the first offering that we had really was inspired by a beautiful crystal structure from Tracy Hall's lab, where they solved the structure of a puff protein. And this revealed that this alpha helical repeat protein had repeats that stacked on top of each other, very much like a stack of pancakes, where each one of these 36 amino acid repeats sandwiched into a nucleotide and its RNA target. And the most elegant thing about the puff proteins is there was a simple code for recognition of RNA bases, where cysteine and glutamine recognized adenine, serine and glutamate guanine, and asparagine and glutamine uracil. However, in natural proteins, there was no code for binding cytosine. And so one of the first things that we did was apply directed evolution to puff repeats to try and create repeats that could bind cytosine. And we did that by randomizing the amino acids at positions 12 and 16 and applying a genetic selection. And from this, we were able to find 5 different combinations, all with arginine at position 16, that could specifically recognize cytosine. And this is just shown in this cartoon from our yeast-3 hybrid system. And we also showed that by swapping the other modules around from different repeats, that they could enable specific recognition of different RNA bases at each position in the protein. And so this gave us a universal code for RNA recognition by puff proteins. We can imagine that these are sort of like Lego blocks, that you can assemble a puff protein with different repeats to target any RNA of interest. And this has been used very successfully by many labs in the RNA world. And I've just got a brief smattering here. And this is by no means exhaustive, but there are some really key players here, such as the Wickens lab and the Wang lab, that have shown that you can manipulate endogenous RNAs at the level of splicing, localization, translation, and stability to create really useful and interesting tools. But while all of this was happening, in our lab, we were really fascinated with mitochondrial gene expression. And we were studying 7 members that existed in mammals of a protein family called the pentatricho repeat proteins. And by making knockouts in mice and in human cells, we were able to characterize these proteins one by one. And I've shown them in green here in the little world of RNA gene expression within mitochondria. And the interesting thing we found about all of these proteins was that they regulate RNA metabolism right from its production, the RNA polymerase, through to processing, modification, and translation. And one of the things that became really clear to us during these studies was that when we looked at their structure, they actually contained a repeating alpha helical unit of 35 amino acids, which was very reminiscent of the 36 amino acids we saw in puff proteins. And we wondered, maybe these proteins could be recognizing RNA in a similar modular way. Except there was a fly in the ointment, because when we were trying to express any of these proteins recombinantly, they were incredibly recalcitrant to this, whether they were our 7 mammalian proteins that we'd discovered and characterized, or whether they were proteins from plants or trypanosomes. The PPR proteins were almost impossible to produce. So we used a little trick that we borrowed from protein engineering called consensus design, where we collected over 20,000 different PPR sequences from NCBI and curated these, and then looked at the most overrepresented amino acids at every position. And then we made an artificial protein, where we made a protein that was consisting of every overrepresented amino acid, and we made an artificial gene that consisted of 8 repeats, by analogy to our puff proteins. And we were really happy to find that these proteins expressed really well in E. coli and were super stable. And even more of a bonus was when we took one of these proteins, our first design, and put it against a variety of homopolymer RNAs, we found that it bound to poly U very tightly and specifically, and not to any other RNA. And luckily for us at the time, there were a number of groups in Australia, Germany, and Japan that were using computational approaches to predict which amino acids and PPR proteins did the binding. And so they predicted the positions 4 and 34 were crucial for this binding. And so we made many different combinations of mutants at these positions within our consensus PPR scaffold, and found one by one that we could find combinations that were specific for individual nucleotides. So now we've realized that we've created 2 universal codes for RNA recognition, both of which can be dialed up simply by changing the amino acids at 2 positions in each of the proteins. However, apart from their universal modes of binding, these are actually quite different when you actually look at them structurally. For example, a puff protein binds RNA in an anti-parallel fashion, with the N-terminus binding the 3' end, whereas PPRs bind in the opposite direction. And whereas puffs bind as a flat arc exposing the phosphate backbone, we were able to solve 4 different structures of our consensus PPRs. And although we couldn't get them with RNA, and when we modeled RNA in, it was a little bit difficult because we couldn't quite join the backbone together, it was pretty clear that RNA would thread down the middle of the protein. So this is quite a different binding mode. So there are different approaches that you can imagine you could use for these, depending on whether you wanted to expose the backbone or enclose the RNA. Then we had one more surprise as well with our consensus PPRs. We were doing a simple control when we took a designed PPR protein that we'd made to target one of our favorite RNA sequences of interest. And as a control, we just wanted to confirm that it was specific for RNA, just like the puffs are. Except we got a bit of a surprise because the equivalent single-stranded DNA sequence actually bound very well. And it wasn't artificial because, or non-specific, because when we looked at a variety of different designed PPR proteins, we could actually get quite specific interactions according to the code we'd determined for RNA. So we realized that there was a specific mode that these would be able to bind both RNA and single-stranded DNA. And at the time, there were no tools to manipulate single-stranded DNA in cells, and we wondered, what could be a cool thing that you could do with a single-stranded DNA targeting protein? And it occurred to us that there's one really interesting place in eukaryotic cells where you have single-stranded DNA, and that's at the end of eukaryotic chromosomes. And of course, telomeres act as sort of a molecular clock, shortening every cell division. apart from in cancer, where aberrant expression of telomerase extends these telomeric single-stranded DNA sequences. And so we thought it would be quite interesting to design our designer PPR proteins to target this single-stranded DNA sequence that was at the end of our chromosomes. This is just showing the results from a telomerase extension assay, where normally you have the telomerase, which is not very processive, stepwise adding different repeats onto the end of the telomeric single-stranded DNA sequence. But if we have specific targeting proteins, we can knock off this activity at really high levels, even on a 1-to-1 molar basis, which would make this the most specific inhibitor of telomerase developed to date. And also it's very specific because you can see our control proteins don't have any effect on telomerase activity. Now, unlike with RNA, we were able to solve the structure both in the presence of a single-stranded DNA and in its absence. And you can see the apo form in light blue, and then the bound form moving there in pink. And so this explains why we weren't able to quite thread through our RNA backbone when we were modeling on our previous structures, simply because there's a massive compaction of about 50% of the height of the protein when it binds nucleic acid. And also, I think this explains quite well how you can inhibit telomerase so potently, because the target is completely encased in protein. So I've shown you two examples of using protein engineering to create new RNA binding proteins. And we've done all of that work using directed evolution in the lab. And this is just a brief summary of how you do these kind of experiments, highlighting that it's actually quite a long and arduous process because you have to make quite large DNA libraries, which are quite diverse and deep. And then you have to create a way of having a life and death selection in a microbe, whether it's a yeast or bacteria. And then you have to do these large, massive screens, amplify the DNA and analyze it. And these can often take many months and a lot of skill and time in the lab. And if you were to have to do this many times, it could take over a year. So we wanted to do something a little bit more extravagant in designing RNA binding proteins and actually evolve an RNA binding protein completely from scratch from a protein that didn't bind RNA at all. So to do this, we took an alternative approach because we knew that directed evolution in the lab would probably be too ambitious. So we decided, why don't we try and create a system of doing directed evolution entirely in a computer? So we skip having to make DNA libraries and just make protein sequences that are mutated directly in the computer, and then select these based on different algorithms, bypassing many of the arduous steps in the lab. And this is just a brief summary of how this works, and I'll step you through this step by step to show you how we did it. So our target we decided to work with is a protein called DHR8. And this is a completely unnatural artificial protein that was designed from first principles by the Baker lab. So it forms a tightly packed alpha helical structure, but has no function at all because it was only designed to form a structure. And what we do is chose residues that were exposed to solvent on this protein to randomize and mask the other structurally important residues, and then made huge libraries of mutants and assessed them by sliding a window along and looking at the properties of each of the amino acids within the 13 amino acid window. And we looked at over 500 different properties, which of course is pretty difficult to condense when you have many, many mutants. And so we used k-means clustering to turn those over 500 properties into a small set of clusters and then into a single waveform for each protein, which enabled us to assess them compared to a library of known RNA binding proteins, which we'd made the same kind of waveforms for, and look for proteins based on this similarity. So every round of evolution, we would take our progenitor protein, our DHR8, create a large number of mutants, and then score them according to the scheme and take the top 10%, and just repeat this again and again to gradually build function within our protein. And so this is what the data actually really looked like. And this is scored based on predicted nucleic acid binding activity by a completely independent means after the run was done. And we've done 3 separate runs, and you can see it's quite able to be reproduced. And you can see that over time, the predicted nucleic binding activity increases until after 20 generations, we reach a plateau. And we took 5 different proteins from these runs and tested their ability to bind RNA. And I've just shown here one group family where we picked 2 of these proteins from, and you can see that this isn't just a random walk through random protein sequence. but there's really a directed evolution happening, and you can see the tree branching and successful lineages continuing until we plucked some of these proteins from the end. And when we looked at the sequences, we saw that they developed regions with some interesting characteristics. They weren't just charged amino acids, but they had a variety of hydrophobic and amphiphilic residues appearing in these exposed sites. And gratifyingly, when we compared them to the progenitor protein that didn't bind any nucleic acid and compared it to our positive control PPR protein that bound poly-A, we could see that these proteins in our electrophoretic mobility shift assays were able to bind nucleic acid, and in fact, they seem to have specificity for guanine homopolymers. And we're currently in the process of characterizing these, and we think these may be a useful addition and form a third family of RNA binding proteins with a universal code. So I hope I've been able to show you today that using synthetic biology, we can create designer RNA binding proteins, and that we hope that these will provide a really useful technology platform to understand natural RNA binding proteins and manipulate transcriptomes. And along the way, we got a little bit of a surprise, and we also managed to create proteins that can specifically bind single-stranded DNA as well. And although we showed that that's quite interesting in terms of blocking telomerase activity, when you think about it, all DNA has to go through single-stranded DNA stage at many different points in its life. Although we think of DNA as always being double-stranded in the cartoons we show the public, of course, when it replicates, is repaired, or recombines, single-stranded DNA is exposed. And we hope that these tools will provide a way to access some of this somewhat neglected regions of biology. So I'd like to thank all the members of my team who have contributed to this work, my collaborators, funding bodies, and of course, most importantly, thank you to you for joining in at all sorts of different and wonderful times around the globe to hear me talk. Thank you so much. And I think we should still have time for some questions. So I'd like to welcome Ron back to help do this bit of discussion.
Thank you, Oliver, for a great talk. And one of the questions that I had was, can you comment on the solubility of the PPR proteins as well as the proteins that you evolved when compared to puffs and their KD values when they're binding RNA?
Mm-hmm. Yeah, so puffs are incredibly tight binders. Many puff proteins are sub-nanomolar binders. Our consensus PPR proteins come pretty close. So there's a bit of overlap, I guess. On average, I'd say the PPR proteins can be slightly less tight compared to puff proteins. Although PPR proteins have an advantage in that while in nature, puff proteins typically have about 8 repeats, PPR proteins are far more variable, and they can have anywhere from 3 to 33 in nature. And so you've got a little bit more scope to modulate the binding affinity than you do with puffs.
Yeah, understood. We have another question from Mingi Shea, and she asked that if the telomerase activity or inhibition of the activity was done in vitro, and does it also work in vivo or in cells?
Mm-hmm. That's work that's still underway, but we have preliminary data that it's looking quite promising. In cells, we've had to experiment a little bit more with targeting, and so we typically fuse these to different protein domains that help them to locate to telomeres, and that seemed to be important to actually get the activity in cells.
Yeah, thank you, Oliver. I think we're kind of up on time, so we'll move on to the next talk. Thank you again, Oliver. And the next up is Dr. Franco Pagani, and the title of his talk is Therapeutic Correction of Splicing Defects Using Modified U1 SNRNPs.
Thank you. Trying to start. What I have to do here, sorry. Can you see? No, I don't know. Can you see it or not?
We can see your slides, Dr. Pagani. It's maybe it's not in the slideshow mode.
Here, it's fine.
Perfect.
Okay, thank you. Thank you very much. So it's really a pleasure to be here, and thank you for the invitation. When we started the project, the idea was to find a way to modulate the spliceosome in a way that it can be used for therapeutic purposes. So this is just a general view of the spliceosome, and we focused essentially on one ribonuclear particle, which is the U1 that you can see here. It's a relatively small RNA, 165 nucleotide, and you have some protein that bind here. And we focus essentially on the tail that it's free to interact with normally with the pre-messenger RNA and with 5' splice site. So we have the gene. This is the gene that code for the U1. It's a relatively short gene. You produce the RNA, then it is assembled, and then it's known to bind to the 5' splice site. The idea was to modify essentially the tail, just change the tail in a way that you can take the U1 and make it binding to other regions. And in particular, we focus to intronic regions just downstream of the exon, because we are interested in finding a way to correct splicing defect, and in particular, exon skipping defect. This is just an example. This is factor 9, coagulation factor 9. It's exon 5. Here we have several disease-causing mutations that induce skipping of the exon. And we take one U1 that bind in this region, we make a screening, and then we are looking at how we can correct. And these are all of these mutations cause exon skipping. They are located at inside the exon, or like this, or they are located in the polypyrimidine tract, or some are located at the consensus 5' splice site. All these mutations are found in patients. And as you can see here, using this U1, we were able to correct a different type of exon skipping mutation. It doesn't work when you have a mutation that are exactly on the dinucleotide that are present on the 5' or on the 3' splice site. So this is working in different systems. These are mini gene experiments. Essentially, what you do is you transfect a mini gene and then you try to correct with the different U1. It works with different mutations. This is just one of the list of the different mutants that we were able to correct. And essentially now I want to focus on two examples. One is very quickly spinal muscular atrophy, in which we have some experience, and then on familial dysautonomia, in which essentially we can also go on and test the effect of the U1 directly in mouse model. Because as you can imagine, when you wanted to do experiment in mice with this type of trick, you should have mice that express the human mutant. So the splicing defect, just a knockout is not sufficient. So the tail is the critical part of the U1, and we started to understand how it works. And this is just an experiment in which we were looking at how is important the tail. We just take, for example, this is acting on spinal muscular atrophy, and this is acting on familial dysautonomia. When you shift the tail, only the tail that is proximal is working. Essentially, when you see here you have the mutant, here the U1 that is working. When you put the tail after, then it doesn't work. So there is the requirement that the sequence of the tail is important near the structure of the U1. The same is happening when you made the other experiment here. Another important point, you can make mismatches and see which are the important positions that are important for the rescue activity. And what was interesting to see here, it seems that you can see that making mutant one by one, the regions that are from position 2, 3, and 5 are the regions that are important for the rescue efficiency. So when you have a mismatch in this position, that start not to work. This is not related to the delta G interaction, but it seems to be more related to some structural requirement between the interaction of the pre-messenger RNA and the U1. It's interesting to know that the position are exactly the position of the canonical 5' splice site when you have the U1. So the second point that we want to address is this is just a summary of several experiments. I don't have time to show all the data, but essentially what we have done, we have tried a way to find the potential of target. What you would expect when you do this type of trick is that the U1 go around and bind on the pre-messenger RNA. So you have potential a lot of binding sites. So you can make a mess on the pre-messenger RNA. And I have to say that we essentially what we have done a very simple experiment. So we take the Philippine cells that overexpress the U1. In this case, one is for U1 and the other is for familial dysautonomia. And we make a RNA-seq analysis once we check that the U1 is expressed. And you see here the effect on splicing on a mini gene. And what was interesting is that we see very few effect on the transcriptome. We see also a very few effect when we looked at an alternative splicing. This is just one example when we looked at skipped exon. If you look, for example, for spinal muscular atrophy, you can see this is the target in the cells, and here are some of the off-target. Obviously, the effect that you see here depends on how you set the threshold of your cutoff. But it seems that the effect is relatively small on, at least in this type of analysis in cells. This is the experiment that we published recently, that I don't go into detail, but this is essentially what we have taken. We have taken the gene, the code for the U1, we inserted in the SMA mice, we inserted in a V vector, and we treat a Taiwanese SMA mice, and then we were looking at different phenotype. And the results are very interesting because it's a systemic treatment. We treat the animals at the beginning of life. And these are the animals that die more or less in 10 days. And when you treat with this AAV that corrects splicing with a U1, we can extend the survival really a lot. So this is our published data. Now I want to show you some recent studies that we are doing on another disease, which is familial dysautonomia. It's a quite complicated disease, but it's a disease that has an effect on the sensory and then the autonomic nervous system. So the idea, it affects different organs, but we were interested in seeing if we can correct also this splicing defect. Most of the patients have a splicing mutation in this position that causes skipping of exon 20. So this has also a tissue-specific distribution in the sense that some organs are more affected than others in terms of exon skipping. So we apply the U1 strategy also in this case. We select some U1 that were active. Essentially, we made a screening, and when we made the screening, we pick up more than one. Then we select one, and then we decide to go on to see the effect on the mice. So we had the opportunity to study these mice. That is a mice from Susan Slagenhout in Boston that she developed. Her group developed these mice. Essentially, you have it's a partial knockout in which you have the insertion of the human transgene with the mutation. So if you can improve splicing, then you expect to correct the defect. So we set up this analysis. We decided to do a particular type of injection. So we made both intracerebral ventricular injection and intraperitoneal injection, because we see that this is the best way to reach most of the tissues, including brain. And we are also interested in reaching the dorsal root ganglia and the sensory ganglia, because it's known that the disease affected this type of cells and tissues. So we decided to do this, and okay, this is essentially are the results. This is the effect on splicing when you treat the animals. So the animals were treated just at P0 and P1, so just at birth. And this is the effect on splicing. As you can see here, we can target with this AAV that express the U1 of specific for FD, we can target different tissues, and this effect essentially depends on the specificity of the AAV. So we can target very well, for example, the liver, the muscle, the heart, but also the dorsal root ganglia, the trigeminal ganglia, and also the brain. You cannot, we don't see a lot of effect on the eye, but the problem is that in the eye, you have a lot of inclusion of the exon. So you started already with a high level of exon inclusion in this tissue. So it seems that it goes where it has to go, the U1. We started, okay, this is just, we are looking now on the protein, but you can see here, for example, in the brain that we can see a rescue in the amount of the protein. there are some very simple experiments that you can do in these mice. So you can study the writing reflex. You can have an effect on the gait analysis and on clasping. And we see a recover when we treat these mice. But the most important result, obviously, is the survival that is ongoing, as you can see here. So you can see here, so this is the survival of this mice. So it has a 50% of mortality after approximately 1 month. And when you treat these mice with the AAV that express the specific U1, you can have a very good recover of the mortality and a very well recover of the survival. So I think that this is my last slide. So the idea is that we can play with this U1 and we can take some targets that are exon skipping defects. So as I told you, not all exon skipping defect can be corrected, but those that, let's say, do not completely disrupt the exon definition, the definition of the exon. Then you do simple experiment on mini genes and you make a U1 screening, and here you can select several candidates that can be tested, for example, for off-target, for safety in relevant cells. Obviously, you want to study human cells that are more similar to the cells that you can target. So you could expect if you want to target the liver, then you have to use liver cells, or if you target brain, to use something that is a motoneuron, that is motoneuron-derived cells. In this case, you select the U1, then you can insert in an AIV, and then the targeting of the U1 in this case will depend on the type of AIV that you use. So there are some AIV that preferentially go to some tissues and not to other. And in that case, then you can treat then, an important point is then to go to a splicing competent humanized animal model, which may be not very easy because there are not a lot of animal model in which the disease is caused by splicing. And at the end, arrive with an AIV that could be used for a personalized therapy to make a correction of specific the defect that is affecting that individual. So I think that I have to thank my collaborator in here, and I have to thank also the Susan Slagenaup in Boston for giving the opportunity to analyze this animal. And the AAV was produced by Janeton, so also the support from different groups. So thank you for your attention, and I'm here to take questions. Thank you.
Thank you, Dr. Pagani, for a great talk. I think we have time for a few questions. So Paul asked if, for the off-target splicing effect, when you make the mismatches, do the off-targets also change?
It's a good question. We have not analyzed one by one, obviously, because that's quite, but this could be an important point. I have to say that when I do this type of experiment, I would expect to see some off target, because obviously it would happen. But we didn't find so much. So that's, and some of the off target that we pick up, we don't understand why they are, if they are really off target or not. So maybe that it's a problem because when you put a cell in culture, they grow different manner. So you are not really looking at a real off target. But that's.
Yeah, sounds good. Thank you. So the next question is from Rhea. She's actually a senior scientist at Locanabio. And she asked, can you comment on the length of U1 5' tail in your off-target experiments?
Yeah, we did. I didn't show you the result. We make it the length more or less. It's a good from 9 to, I think that the best length is 16 nucleotide. When we change the length, so we make a systematic studies to see with the length, and more or less 15 nucleotide is the best. At least in two systems that we tested, is the best length.
Yeah, understood. She also asked, I think it's a really interesting question. She asked if you have seen how much of the synthetic U1 that you're making gets assembled as SNRP.
It depends on the experiment. We were looking at, we make the ratio between the U1 that we synthesize, the U1 that we introduce, and the endogenous U1. And depending on the AIV, depending on the experiment, it goes from less than 1% to 10% of the total U1. Well, I didn't show the experiment, but we have an experiment in which we have we overexpress from the transgenic mice the U1 for the spinal muscular atrophy, and it is less than 1%. And when you cross these mice with the mice of SMA, you recover the splicing defect and you recover the phenotype. So it seems that the therapeutic level of this U1 is quite low. So even if you have less than 1%, then you still have an effect.
Yeah, makes sense. So I actually have one or two questions. My first question is, I'm assuming that you're using the U1 promoter so that you can get proper processing. Have you tried other promoters at all?
No, we have not tried. The important point is that we take it like a block cassette, so we cannot modify it a lot, because obviously you have to consider that the promoter, the start of transcription should be exactly when you expect to be, because if not, the tail of the U1 will change accordingly. So it seems that we have never tried to do, but you cannot move some of the regulatory element in the promoter, because then you can have an effect on when it starts transcription. And so the RNA will be different.
Yeah, understood. My next question is around the dose of AAV that you were using, and if you're injecting in the neonatal mice or adult mice.
We are injecting in neonatal mice, and was the maximal dose that we can reach. It's quite high dose, I have to say that. But the problem is that these mice are very sick, so you need to have and you need to have a strong effect just in the few weeks of life, because probably these are the window in which you have to treat the mice in order to have a rescue of the phenotype. So if you wait one week, then probably you will have no effect.
Got it. And last question, in the panel, and I'm paraphrasing, do you always have like Agu-Ragu sequence as the target sequence? And how important is the distance between the target to the 5' splice site?
The importance of the distance depends on the context. In some context, in the spinal muscular atrophy and in FD, till 50 nucleotide downstream, the 5' splice site, they are still working, then start not to work. In other cases, it depends. You have only a narrow window. We don't know if this depends on the consensus on the sequence, because maybe that the sequence, so we don't know if the U1 are produced or not. Maybe that the U1 that when we made the screening, maybe that the tail that we change it is, for example, GC rich, and maybe that the U1 is not assembled in that case. So.
Sounds good. I think we have time for one more question. And this question is, could off-target effects be reduced by reducing concentration of the delivered U1 while keeping the specificity?
We have not checked at this point, because we prefer to have a high expression level and to see the maximum, because that cells express more or less 20% relative to the endogenous U1, the cell that we tested. And these are stable clones. So we inserted 3 copies of our U1. So I would expect to see less off target, but we have not done this in a systematic way, because we need to spend a lot of money to do RNA-seq in this.
Yeah, totally.
For the mice.
Have you seen any toxicity at like some of the high doses at all?
No, we have not seen in the mice, but we didn't check. So we have not done the classical experiment that you have to do to see toxicity in the mice, essentially. So we have not done this experiment.
Sounds good. And thank you very much again for your talk, and to the panel and the audience for great questions. So time to move on to the next talk. And I would like to invite Mitch O'Connell from University of Rochester. And the title of his talk is Insights into RNA Binding and RNA Cleavage Specificities of CRISPR-Cas13. Thank you, Mitch. Welcome.
Thank you. Thanks for the invitation and good morning, afternoon, and evening to everyone. And everyone can see my slides. Good. All right. So today I'm going to tell you about some of the work we're doing, really kind of trying to dig into understanding how these CRISPR enzymes are able to bind and recognize RNA and then undergo some kind of conformational change in order to activate their nucleases. And so my lab is broadly interested in kind of two aspects of RNA. We do a lot of work on RNA biology, trying to understand a particular class of RNA modification enzymes, but we also spend quite a lot of time trying to understand RNA targeting CRISPR systems, both from their pure enzymology to applications development. And today I'm going to be primarily focusing on Cas13. And so, as many of you know, these CRISPR systems come in many flavors, and they can target RNA. So anything from these very complex class 1 systems, such as the type 3 systems that can target RNA, to the much more abbreviated class 2 systems, such as Cas9, which we'd previously shown can target RNA under some circumstances, and more recently Cas13, that seems to be a bona fide RNA targeting single effector protein. And so for us, beyond the super interesting CRISPR biology and the enzymology, it's also an exciting opportunity for programmable RNA targeting tools. And so just jumping straight in, based on a lot of work by many people, we've now figured out how this enzyme Cas13 works, and we consider it a guide RNA programmable target RNA activated non-specific RNAs. And I'll show you what I mean by that. And so we have this large single polypeptide, often over 100 kilodaltons. It contains these two characteristic Hep N domains. And so these come together in 3D space to form a composite active site that in the apoprotein is not active. This protein is able to bind a pre-CRISPR RNA and actually process it using a separate RNase active site that's different from these Hep N domains to form this binary complex. And it's upon target RNA recognition and binding that you get a conformational change within these Hep Ns that lead to an active site that's capable of cleaving RNA. And what can happen here is you end up with either what we consider cis RNA cleavage, where based on the crystal and cryo structures that we have, this active site's kind of solvent exposed and away from the target RNA guide duplex, and you get cleavage in cis if the RNA is long enough and can, oh, I've just got a fire alarm. That's not useful. Not really sure what to do here.
I think you should exit your building to be safe.
Yeah, I could probably try and, if you give me like 1-2 minutes, I could run downstairs and hopefully have enough internet to continue. I'll do that. I am going to, sorry about this. This is not common.
Never boring. Hi, everyone. It's never boring, so I guess we can enjoy the break and answer any unanswered questions so far. So please keep posing your questions in the Q&A. One of the questions for Dr. Pagani, did you say that you inserted 3 copies of U1 in your construct?
In the flip-in cell, yes, we inserted 3 copies, just to have a high expression of the U1.
Understood. Have you tried exon skipping assays with U1 as well?
Yeah, it didn't work. In our hands, it didn't work. So it doesn't work like an antisense. It works like a recruiting splicing factor, we think.
Sounds good. Oliver, have you tried to package any of the PPR proteins or your new RNA binding proteins in AAV for expression in different tissues?
We haven't personally done that in my lab, but I know labs that have packaged them successfully in AAV. So, and I think that's one of the advantages of some of these proteins compared to, say, the larger Cas proteins, which, as Mitch so wisely pointed out, are often over hundreds of kilodaltons. Whereas the puff proteins and PPR proteins are actually relatively small, starting around 30 kilodaltons. So they're actually very well suited for AAV delivery.
Understood. You know, one of the questions that keeps coming up, and a question for CRISPR-Cas13 is their off targets. So when you're looking at binding, have you looked at off targets for some of these proteins, and what is kind of the specificity for the target itself?
Yeah, so the specificity can vary depending on the protein, which is quite interesting. So there are some puff proteins that when you look at off targets, they're incredibly specific. They only have a handful or tens within a transcriptome, where there are some that have a lot more. And so I think the nature of these proteins is you need to tweak a little bit the sequences that you're targeting, because because they have much higher affinity than a nucleotide-nucleotide interaction that you have in the context of a Cas, that there's an ability to have either very low off targets or very high, depending on the sequence. So it's really a matter of finding your best one for your target.
Got it. So another question for you, Oliver. This is Angie from Locana. You probably know her well. And she asked, with your new computation method for protein evolution, would it be possible to engineer the PPR to be specific for RNA and not bind a single-stranded DNA?
Yeah, that's something we're interested in, and that's because we have structures now of our consensus PPRs, and that's something we're very interested in doing, and we're actually actively working on at the moment. I think our computational evolution program is actually, we sort of view it as something that's suited for more ambitious aims, because you can really perform, as I showed in some of my examples, you can do 20 rounds of evolution in less than 24 hours. That's why we chose to take something a lot more ambitious, a protein that has no activity, and turned it into a protein with activity. And so we see the computational approach is really for these kind of ambitious things where you want to create a complete new function or a new binding specificity, rather than sort of more tweaking type things, which might be more suitable for traditional directed evolution.
Got it. And I guess I'll ask this question loudly, Dr. Pagani. This is for you. Are there any limits in terms of what exons you can correct? For example, very short exons, very long exons, or an exons following an intron or an intron definition model?
Oh, thank you. We tried with short, we have no problem. We have found with long, we have not tried. Essentially, it's the context that made the difference. There are some cases in which we were not able to work, and that it depends on the fact that there was a strong silencer inside the exon. So that make the big difference. So.
Sounds good. One question for you, Dr. Pagani. Have you tried to include any cryptic exons?
No, we have not tried. But it should work. It should work, I think, depending on how strong is the definition of the exon.
Yeah, that would be a good method to knock down a gene.
Yeah,
like that. While we're still waiting for Mitch, I just want to remind everyone that right after this talk, RNA Society will continue with their concurrent session, so don't miss that.
Well, good morning, afternoon, evening to everyone that may listen to this. I apologize for my disappearance earlier. We had a fire alarm and it was a bit chaotic here, and I was unable to find a spot to continue the talk. So sincerest apologies, and I'm sad to have missed the rest of the session and the discussion, but in order to try and make up for it, I've recorded this and I'd be happy to take any questions via email if you have anything you'd like to ask me. So my talk was meant to be about kind of a small part of our work looking into the insights into the RNA binding and cleavage specificities of CRISPR-Cas13. And so broadly, my lab works on two major areas. We've become increasingly interested in particular RNA modifications and how they are deposited on particular RNA transcripts. And the other part of the lab's really interested in not only the biology, and increasingly more so the biology of RNA targeting CRISPR systems, but also how to develop them as new and improved RNA targeting tools. And kind of in order to get there, we're taking a kind of reductionist approach and really trying to understand how these enzymes work and the quirks of their specificity. And today I really want to talk to you about that in particular, binding versus cleavage, and the kind of requisite steps that need to occur for this process to occur, and how specificity can modulate this. And so, as many of you are probably aware, CRISPR-Cas systems come in many flavors and increasingly so, and several of them, these types of CRISPR-Cas systems can target RNA. So we have these very complicated class 1 systems with multi-subunit complexes. And for example, the class 3 system can target RNA. And then we have these much more abbreviated class 2 systems, including Cas9, which we've shown previously under certain circumstances can target RNA, and Cas13, which I'll talk more about today. So this enzyme we like to think of as a guide RNA programmable target RNA activated non-specific RNAs. And sorry, my face gets in the way of the title there, but the way this works, you have this single polypeptide that has these two HepN domains that form this composite active site in the protein. This protein is able to bind a CRISPR RNA, a pre-CRISPR RNA, and then using a separate nuclease domain within the enzyme is able to cleave this to produce a product-bound mature binary complex comprising a guide RNA and the protein. Upon sufficient complementarity, you can get stable RNA binding and activation of the HepN nuclease. And so there's this subtle conformational change that brings the active site residues to form this composite pocket that's very solvent exposed, and this can result in kind of two major RNA cleavage activities. We have this cis RNA cleavage activity. If the RNA is long enough, naturally it's going to get cleaved by the active site. What's worth noticing here is that this active site's really not facing down and interacting with this guide target duplex, but rather is very solvent exposed. And this enables a different kind of activity that was a surprise to many of us when we first started working on this enzyme, known as transcleavage or collateral cleavage or even bystander cleavage RNA activity. And interestingly, this has been harnessed as a very sensitive diagnostic by many folks out there. And also this activity is rampant in the bacterial hosts that these enzymes emanate from. Interestingly, not a lot of evidence for transcleavage in mammalian cells, although as we saw from some talks in a later session today, there is some evidence depending on target transcript levels. And that's a phenomenon that I think really needs further exploration. But for now, when targeting endogenous genes, it seems like this is a really robust RNA targeting tool. And so we're really interested in this kind of process here in terms of what's required for stable target binding versus activation of this HepN nuclease. And the reason we're interested in this is because we could use this RNA-guided protein as not only a nuclease to cleave particular RNA transcripts, but also by inactivating the nuclease domains, you can imagine a number of other applications where you're modulating various aspects of RNA biology. So the question for us really becomes, as some of these applications rely on RNA-guided RNA cleavage potential of Cas13, while others rely on RNA-guided RNA binding potential Cas13, are there differences in the specificity profiles of these two activities? And so really schematized here, you can see that a loaded Cas13 searching for a target, it has a particular affinity for binding to a target RNA. And the question is, can it bind stably to these target RNAs without activating its nuclease, or is this process kind of coupled? Or are there additional kind of proofreading steps where you need additional base pairing interactions? And so that's really the question we're kind of trying to answer. And so thinking about this, the first thing is, what do we actually know about Cas13's target RNA binding specificity? And by that, I mean, if you introduce mismatches in target or in the guide, how does that affect the affinity for this enzyme for a particular target sequence? And really, we didn't know anything at the time. And the next question is, well, what is known about, I guess, what we could consider nuclease activation specificity? So how do mismatches affect the nuclease activity of this enzyme? And there are several publications out there and studies that have kind of begun to understand this. And what we noticed is that really the different regions of the guide RNA, target RNA duplex are more sensitive to mismatches. And this can often inform you how to design better guides and where off targets could be coming from. And so what we really notice is that different types of Cas13s from their different subtypes have kind of different sensitivities to these mismatches, not only in region, but also amount. And so the question really is there additional proofreading beyond stable binding? And the reason we're really interested in this is because there's a precedent in nucleic acid guided proteins where this is the case. And so a classic example is work done on Cas9, where you can trap Cas9 will bind to certain double-stranded DNA sequences with a relatively high affinity, but unable to cleave them with much efficiency. And it's not really, and what was shown is it's not really able to activate its nucleases until there's base pairing to the distal region to the PAM sequence. So if you have the PAM here, it's really the base pairing at the PAM distal end of the target guide duplex that is activating this concerted conformational change in cleavage by the RUVC and the H&H domains. And so really you can get stable binding without any, without robust cleavage activity. And the same is actually true for Argonaut proteins. And what's interesting there is actually pretty substantial differences between different orthologs of Argonauts, and we're kind of curious if the same thing applies for Cas13s. And so in this case, from this really nice study here, you can measure really high and quite tight binding affinities for mismatched targets, but these are not competent to activate the slicer activity of Argonaut. So it's really this kind of microRNA versus siRNA pathways. And what's really interesting is the binding's incredibly tight, but there's absolutely no enzymatic activity. And so for us, we really just, using these as examples, want to understand whether we can kind of tease apart whether you can obtain stable target binding with Cas13 versus the activation of the nuclease. And so for this first and most of the talk, I'm going to be talking about Cas13A, and later on I will show you some very hot off the press data on Cas13D, which shows some interesting similarities, but also differences in the way that they are affected by mismatches in the guide target duplex. And so without going into too much detail, we developed a very Cas13 specific RNA bind and seek approach to test the effects of mismatches in a kind of high throughput manner using next generation sequencing. And what we're looking at here is a matrix of all pairs of mismatches. And what I want to tell you is that if you look at single mismatches just look at all the subsets of library members that have single mismatches in data that I'm not going to show you, it's really the middle of the guide that is the most sensitive region to mismatches. So in other words, if you deliberately introduce a mismatch in that region, you lose a substantial amount of binding affinity. And so what was interesting here is when we start looking at pairs of mismatches, indeed, the center of the guide really does pop up as the most affected region, shown here in blue. But what's interesting is that you compare some of these very heavily affected mismatches in the central region with other mismatches in a separate region, and you actually see somewhat of a recovery of mismatch sensitivity. And the reason we think this is, that we believe this, is because you can make mismatches outside in the flanking regions coupled with mismatches in this super sensitive region, and you still see a big loss in affinity. But somehow a mismatch in this 4 to 8 region is able to somehow recover some of that affinity, at least in this experiment. And we thought that was an interesting quirk of the enzyme, and we wanted to explore that further. So we went back to some classic filter binding assays and were able to show, just jumping to the punchline, that indeed, while mismatches directly in the middle of this guide target region leads to a large loss in affinity, greater than 10 to over 1,000-fold. It's not even measurable in some cases, depending on which RNA sequence you do this to. But if you just move these mismatches slightly more towards the 3' end of the target in this 5 to 8 region, you actually see an increase in binding affinity, so a tighter binding event. And this was a little surprising to us, but kind of mirrored what we'd seen with the argonauts proteins with this weird mismatch enhanced affinity. And so the next question was, we had these odd substrates that are mismatched within the duplex, but are able to bind incredibly tightly. Are they still able to activate the enzyme for RNA cleavage, this HepN conformational change? And so here, and this was done in collaboration with some excellent graduate students in Jennifer's lab as I was starting my own lab, and getting up and running here in Rochester. And what we showed was that indeed mismatches in that central region, as expected, you have really poor binding, so naturally you're not going to get a ton of RNA cleavage. But what was interesting is that this 5 to 8 region, again, while binding incredibly tightly to the to Cas13, we couldn't detect any activation of that HepN nuclease. And this is different compared to different parts of the other mismatched regions of the guide target duplex. So for us, that was super surprising. Again, really tight binding, but absolutely no activation of the nuclease. And looking at and gleaning at a lot of the cryo and crystal structures that have come out over the past couple of years, and particularly this cryo structure from Yang-Li Wang, is that there's a region that's actually sensing the target RNA, guide RNA duplex in this region consists of this alpha helix that is in very close spatial proximity to the HepN active site. And one could really imagine that the ability for this helix to lay across and sense this duplex could really affect whether this conformational change could occur. And so any disruption in the orientation of this helix would naturally maybe prevent activation. And this is something we're exploring further. And we really think this might be a high energy barrier that somehow evolution has incorporated into this protein to prevent spurious self-targeting. And so a lot of the initial target RNA binding is probably driven by simple hybridization kinetics, but the enzyme needs to evolve a way to know that it has a very very close to perfect complement of a target before it's going to activate. We see this in a lot of these other enzymes as well. And so I think it's a balance. It's a kind of strategy that evolution's come up with to control these enzymes, given the barriers of hybridization kinetics. And so with that, you can come up with a model. I won't go too much into the details, but it's really that the take home here is that there is a differential specificity and I guess plasticity for stable RNA binding versus stable activation of the nuclease. And this really needs to be considered when designing guide RNAs and thinking about off targets for those various applications. And thinking about this some more, there might be ways to carry out protein engineering efforts for more specific Cas13 binders. And this is something we're thinking about a lot in the lab. And finally, there might be some interesting evolutionary constraints with this as well. And thinking about this in terms of prokaryotic immunity and the role of binding a target versus cleaving a target. And so just in the last couple of minutes, I'm going to talk very briefly about some similar work that we've done on RFX Cas13D, asking similar questions. And this has really been spearheaded by a talented graduate student in the lab, Adri. And what he's done is basically taken a similar approach where we've made a bunch of mismatched targets or mismatched guides and asked how do they affect binding affinity. And very, very new data that we need to replicate, we show that indeed mismatches affect binding affinity. But interestingly, the affinity is not affected as much as we would expect across the board, sometimes only a 50% reduction. And interestingly, there's certain areas of the guide RNA that don't seem to be affected by mismatches at all. And the stem disruption is a variant direct repeat that we're studying based on some work from Neville Sanjana's lab. And it seems like we're trying to figure out why this stem disruption it leads to higher targeting in mammalian cells and what the biochemical basis for that might be. And so far it's behaving pretty well in our hands. And so the next question is, if you do the same experiment where you're looking at RNA cleavage specificities and activation of the heparin nuclease, we see something a little different to what we saw with the Cas13A that we were working on. And so if you put these side to side, we saw a loss in binding affinity, in some cases pretty substantial, in some cases not so substantial. But then when you look at cleavage activity, it seems across the board that this enzyme is a lot more sensitive to mismatches between the guide and target region and leads to a larger loss in affinities, suggesting that this enzyme is a little more specific in terms of heparin nuclease activation. Interestingly, again, it's a region that binds tightly is also activated pretty well too, even with substantial mismatches. So in terms of guide design, this might be a particular region to pay attention to, much like the pan-proximal region for Cas9 when we design guides for gene editing experiments. And so in conclusion, we know that Cas13 acts as what we consider an exoRNA sentinel. When activated, results in global host RNA degradation, but also cell dormancy upon phage or memogenetic element infection. And so in bacteria and the test tube, this is kind of its role. And we know in mammalian cells, there's some evidence that a lot of evidence that it works as a really nice RNA knockdown tool when targeting endogenous genes. And much like Cas9 and the Argonaut proteins, we see this decoupled RNA binding and RNA cleavage specificity. And really, I think this is a balance that the enzyme's trying to play with the strength of hybridization kinetics versus controlling a nuclease that could be quite toxic to the cells. And so, interestingly, very preliminary data, but we think different orthologs appear to have different RNA binding and cleavage specificity, and this needs to be, we're trying to develop approaches to test this more thoroughly in a more high throughput manner to really get out, to figure out which ones are best for applications. And so to finish, I'd like to thank everyone in my current lab that's working on this and many other projects related to CRISPR biology and applications, everyone in my postdoc lab that helped in this kind of transition period with this Cas13A stuff we were working on, collaborators past and present, funding sources, and I have two disclosures here as well. So unfortunately, I won't be able to take questions here because this is pre-recorded, but feel free to email me if you ever get around to watching this. And again, sorry for the fire alarm.